Thank you very much, Patricia, for the nice introduction, and thank you for the invitation. I wish I could be with you in person, but of course, under the circumstances, it's not possible. My last visit to Brazil goes back almost 15 years, and I wish I could be there again. There's another reason why it's a pleasure for me to talk to you today. I realize that the topic of my presentation is not necessarily perfectly aligned with veterinary research. However, parasites are important for all living organisms. And I have spent my entire career trying to convince other biologists, especially ecologists, evolutionary biologists, and people who study animal behavior, that parasites are very important. Unfortunately, I have been facing one particular problem, and this is that for most people, little things don't matter. Most people think that it's the big organisms in nature that are the most important in ecosystems and in the functioning of natural communities. And of course, parasites are located at this end of the spectrum. They tend to be very small. And yet, of course, this particular bias that people have is completely false. Small things can have disproportionately large impact on natural systems. And this is particularly true in the case of the impact of parasites on animal behavior. Perhaps the book that has been the most influential for me is this book by Richard Dawkins, published in the early 1980s, called The Extended Phenotype. In this book, Dawkins argued that genes inside one organism could have impacts on the phenotype of other organisms. And one of the best examples he could use was to say that genes inside a parasite could affect the behavior of their hosts. Now, this was quite a revolutionary idea at the time. There were just a few examples that parasites were perhaps capable of taking over the behavior of their hosts. But of course, since then, there's been a lot of research that has shown that this is not just possible, but it actually happens in many, many cases. And I have spent a lot of my time over the past 30 years working on this particular topic. So today, what I would like to do in this presentation is first of all give you a primer, an introduction on host manipulation by parasites, focusing in particular on the types of parasites that are capable of manipulating behavior. Secondly, I would like to argue that the impact of host manipulation by parasites extends beyond their immediate impact on the host, but that their impact can affect the way we study animal behavior, but also can affect the functioning of natural communities. And finally, I want to discuss some of the recent work we've been doing, trying to elucidate the mechanisms by which a parasite can modify the behavior of its host. But first, just a brief introduction to the concept of host manipulation by parasites. Natural selection will favor parasites that can change the behavior of their host if it can improve the parasite's transmission success. And there are 4 types of life cycles where there are many examples of parasites that can alter host behavior. They are summarized on this slide, but let me discuss them one by one. And the first is vector transmission. Many parasites are transmitted by vectors, such as blood-sucking mosquitoes, for instance. And there's a lot of research now showing that the biting behavior of these vectors can be altered so that they take shorter meals on each host, but visit more hosts per unit time. And this, of course, can only improve the chances of parasite transmission. This has been shown to apply to the vectors of Plasmodium, the agent that causes malaria, but also a range of other parasites, such as viruses and filarial nematodes. Other research has also shown that the smell, the odors produced by infected vertebrates are different and therefore attract more mosquitoes. So in this case, these vector-transmitted parasites can manipulate the phenotype of one host and the behavior of the other host in order to improve their transmission. Another type of parasites capable of doing that are the parasitoids. Now, parasitoids, if you don't know, are small insects, often wasps, that lay their eggs inside other insects. And their eggs then grow into larvae inside the insect host until they come out and they pupate attached to vegetation. Now, many of these parasitoids can manipulate the behavior of their host to cause this host to defend them against predators. For instance, in the video shown here, all these little white structures on the little branch here are the pupae of a wasp parasite that used to infect this caterpillar. Now that they have emerged from their host, they're attached to the vegetation, but they are still capable of controlling the behavior of their host so that their host stays behind and will defend them against predators. For instance, it is currently moving around and trying to knock out a little insect trying to crawl up to eat the pupae. So this insect is defending its former parasites. They're not even in its body anymore, but its behavior is still programmed to defend the parasite. And there are many other examples like this. For instance, the little beetle here is protecting the pupa of a wasp that used to be its parasite. It will also protect it against predators. So that's a second type of life cycle where natural selection has favored this ability to take control of the host behavior. A third type of life cycle where this phenomenon is common includes life cycle in which the host must switch to a different habitat completely in order for the parasite to continue its life cycle. So at some point, the parasite must leave the body of the host into a microhabitat that is very different from where the host normally lives. And the parasite forces the host to go to this microhabitat. This is common, for instance, in myrmitid nematodes, which infect terrestrial arthropods like this little amphipod or this earwig. The parasites can grow to very large sizes inside their host. And when they're ready to come out and live their short adult life, they must cause their host to find water, or at least water-saturated substrate, because the parasites are very prone to desiccation. And what has been found in these parasites is that when they're ready to come out, the behavior of their host changes completely, and they start looking for water or water-saturated soil or sand, and they will stay in this water-saturated habitat until the parasite comes out. Perhaps the type of parasite that has been the best studied in this context are nematomorphs, which are not nematodes. They belong to a completely different phylum known as the Nematomorpha. But they also grow inside the terrestrial host, like this cricket. And when they are ready to come out, they force it to find water. And you can see there the cricket just jumped in water. This is a completely abnormal behavior for a cricket. If you were to remove it from the swimming pool, it would keep jumping back in. And this is because the parasite has taken over control of its behavior and it wants to go in the water. When it's in the water, as you can see, the parasite is now coming out of the cricket. The size of the parasite is huge, so the internal organs of that cricket have been damaged, and it is unlikely to survive this particular event. But for the parasite, reaching water is essential because this is where it needs to find a mate, produce its eggs, and then the parasite will die. So in these sorts of life cycles, it is common for the parasite to take control of the host, make it go to a place where the host would never go. And this, like we've seen, is seen in these nematomorphs, but also in the nematodes, which belong to a completely different phylum, but have convergently evolved a very similar life cycle. But the final type of life cycles in which host manipulation is common is probably the one that has been studied the most. And this is life cycle that involves trophic transmission or transmission by predation. So in these life cycles, the parasite uses an intermediate host and must be transmitted by predation to a definitive host. Obviously, many of these intermediate hosts have evolved anti-predator adaptations. So the parasite must change their behavior so that they become more susceptible to be eaten by their next host. The classic textbook examples of this is the trematode dicrocilium, which at some point in its life cycle must be transmitted from an ant to a grazing herbivore like a sheep. But of course, sheep do not feed on small insects, they feed on grass. So what the parasite does is that it attaches to the back of the brain of the ant, and then it causes it to climb to the tip of a grass blade, bite onto it, and stay there for many hours at a time, awaiting a possible chance during a chance event during which it might be ingested by a grazing herbivore. Another completely different example involves the well-known parasite Toxoplasma gondii, which has to be transmitted from a rodent intermediate host to a cat definitive host, again transmitted by predation. Obviously, rats have evolved a range of adaptation to avoid cats, but some research shows that when rats are infected by Toxoplasma gondii, they are suddenly attracted to the odor of cats, which is something completely unnatural for a rat or a mouse. Normally, they would avoid the odor of cats, obviously. But in this case, the parasites has altered the sensory perception of its host to improve its own chances of transmission. It's not just the behavior of the intermediate host that might be changed, it could also be their appearance. For instance, in these little crustaceans shown here, you can see that there are different color morphs. They range from pale brown to green in coloration. And yet all of these belong to the same species. And what happens is that these crustaceans harbor the juvenile stages of acanthocephalans, you have one shown here, but also of cestodes. And when you look at the proportion of individuals that are infected by these different parasites, you find that it is associated with their color. So what we have here is the different color morphs of these particular crustaceans. And you can see that the ones that are dark gray or green have a much greater chance of harboring the acanthocephalan parasites. In contrast, the ones that are green or blue, which is these two types here, have a much higher proportion, a much higher chance of harboring cestode parasites. So in this case, these parasites can modify the color of their hosts to make them more visible against their background and facilitate their transmission to their next host, which happens to be shorebirds like seagulls, for instance. There are even more spectacular examples of parasites altering the appearance of their host. Consider, for instance, the species of nematode, which lives in Central America and which must be transmitted from an ant to a bird. But unfortunately for this parasite, the birds that are suitable for its development do not feed on insects, but they feed on small fruits, small berries. So what the parasite does is that it changes the color of its ant. The posterior end of the ant turns from black to a bright red color, and it also changes the behavior of the ants, forcing them to go and perch themselves motionless for many hours at a time in patches of small red berries. So in the photo that you have here, all these red structures are small fruit, except this here. And you can see that this part of the ant now matches very perfectly the size and coloration of these small fruit. And presumably birds are being fooled by these mimics, and they eat the ant thinking it's a fruit, but instead it's just a bunch of small nematodes that will infect it. And as a final example, the most spectacular example of this phenomenon involves the trematode leucochloridium, which must be transmitted from a snail to a bird. But again, the problem for this parasite is that the birds that are suitable for its development don't feed on snails, but they feed on caterpillars. So what the parasites do is that after they multiply asexually within the snail in structures that are called sporocysts, these sporocysts will move into the tentacles of the snail. They cause them to become stretched so thin that they become transparent, and we can actually see the bright colors of the sporocysts inside the snail. And these sporocysts are also capable of pulsating. They move back and forth, creating the appearance of a moving caterpillar. And again, this great imitation presumably fools a bird into feeding on that part of the snail, which it believes is a caterpillar, but in fact is just a bag full of parasites. So you can see there are many examples of parasites that alter the phenotype of their host, whether it's the behavior or the appearance. And over the years, the number of examples has grown dramatically. This graph shows the growth in the number of studies that have focused on behavioral manipulation by parasites. And this graph only includes data on parasites transmitted trophically. If we included all parasites, there would be even more examples. But what the gray line shows is the cumulative number of published empirical studies that have demonstrated this phenomenon. As you can see that it's been growing extremely rapidly. The blue and the red lines show instead the number of different parasite taxa or different host taxa that have been shown to be involved in cases of host manipulation by parasites. You can see that these are also growing over time, but at a much slower rate. And that is in large part because in the past 10 or 15 years, several researchers have focused their research on a few model systems, such as those two, for instance. So many people work on these species only. This is a common phenomenon in science where over time people adopt model systems and start to know a lot more about these model systems, and therefore the depth of our knowledge increases, but the breadth of our knowledge doesn't. So we focus our attention on a few model systems. So this has disadvantages. We compromise how well we know the phenomenon versus how broadly we know about it across many species. This concludes the first part of my talk. I now want to try to emphasize to you that this phenomenon has implications that go beyond just parasite transmission. The ability of parasites to change the behavior of their host is not just something of interest to parasitologists. It has broad impacts on the study of ecology and the study of animal behavior. And as an example of how this phenomenon can impact ecology, I just want to summarize a series of studies we have done many years ago on marine ecosystems just a few kilometers from my office. And these studies focused on parasites of a bivalve, the New Zealand cockle, which is an extremely common bivalve in intertidal areas around here. This bivalve is the host of a trematode, Curtitaria australis, which lives as an adult inside oyster catchers. The eggs of these parasites pass out in the feces of the bird. Once on the sediment of the intertidal zone, the larvae that come out of the eggs will infect the snail, multiply asexually within the snail, and the little infective stages that come out, known as cercariae, you have them here, you also have a close-up view of one right here. These will swim around until they get sucked into the inhalant siphon of the bivalve, and then they encyst in the bivalve and they await ingestion by a bird. If they're lucky, they will be eaten by a bird and complete their life cycle. When they are inside the bivalve, they do not encyst anywhere in its tissue. They encyst specifically in the foot, which is the muscle that the bivalve uses to burrow into the sediment. And you can see that they can accumulate large numbers inside that foot. And at some point, when there are many of these parasites inside the foot of one bivalve, they start to replace the the muscle tissue inside the foot, so that the bivalve is no longer capable of burrowing. And this results in a population of bivalve which is split into two types. The ones that have few parasites are still capable of burrowing, and they disappear a couple of centimeters below the sediment surface. But the ones that have a lot of parasites inside their foot are no longer capable of burrowing, and they remain visible at the surface of the sediment. We have shown with field experiments that this makes them much more susceptible to predation by birds. However, the implications of this change in the burrowing behavior goes beyond transmission. When you have many of these bivalves at the surface of the sediment, it causes a change in the habitat in two different ways. The first is that the presence of these structures affects the flow of water and the pattern of sedimentation, so that you get much finer sediments that deposit in these areas. And this is very important for habitat selection by a range of invertebrates that will choose whether or not to settle in a particular area. But also the presence of these bivalves at the surface provides a hard substrate for which other invertebrates can attach, and it also provides shelter to some other smaller invertebrates. So we tested the hypothesis that perhaps the presence of these bivalves at the surface, which is only due to the parasites, may affect the types and the number of invertebrate species that settle in areas where you have a lot of parasites and therefore a lot of these bivalves at the surface. So we ran a 1-year-long field experiment where we used experimental plots in which we simulated few parasites or no parasites or many parasites by having either no bivalves at the surface, a few or many bivalves at the surface. And what we found after 1 year, when we sampled these different experimental plots, was that the diversity of organisms that had settled there was very different among them. The bivalves, which I should have said are called cockles, that's the name of the bivalve species. In these experimental plots, there were either no bivalves at the surface, there were a few, or there were many. So this simulates areas without parasites, a few parasites, or many parasites infecting the bivalves. And we took core samples of the sediment, which we then passed through a sieve to find all the little organisms living in it. And we found that there was significantly more species of invertebrates living in the areas where we simulated the presence of the parasite than in the areas where we did not simulate the parasite. And the data here is expressed as the number of invertebrate species per core sample. Now, these core samples are just cylinders that we drive into the sediment, and then we remove the old sediment. They are about the same size as a can of Coca-Cola. So it's a very small amount of sediment. And on average, there was 3 extra species in these samples in areas where there were many bivalves at the surface. So the point of this story is that one parasite species, which affects the behavior, the burrowing behavior of one host species, in this case affects the biodiversity of the entire intertidal ecosystem. There have been a few other examples like these, but it shows that these manipulative parasites don't just affect the transmission of the parasite, they can have indirect cascading effects on the entire animal community. And manipulative parasites can also affect how people study animal behavior and what sort of behavioral measures we take. In the last 15 years or so, one of the most common or important framework to study animal behavior is this concept of animal personality. The idea that individual animals in one population have particular behavioral patterns that make them slightly different from one another. And people who study animal personality take 2 particular approaches to study it. The first is to demonstrate that there is behavioral consistency or behavioral predictability in particular animals. In this little example here, for instance, if each symbol represents a different individual animals, and if each individual animal has its behavior measured on 3 separate occasions, the existence of personality would mean that we would find more consistency in the measurements taken on the same individuals than if you look at the measurements taken across the whole population. In statistical terms, it would be that the variance within individual is smaller than the variance amongst individuals. So animals have a consistent behavioral pattern that you can measure with similar values on different occasions. The other characteristic of animal personality is that if you measure two different behaviors across many different individuals, you tend to find correlations between these behaviors, something that people have been calling behavioral syndromes. And in this case here, we have two behaviors, Y and Z, that have been measured in several different individuals. And if there is the existence of personality features in this population, you would expect that some animals that measure, that have a high score for a particular behavior would also have a high score for a different behavior. And those that have a low value for one behavior may also have a low value for a different behavior. So this approach has been used by animal behavior researchers in the past 15 years to characterize the behavior of wild animals. A few years ago, I've emphasized in a publication that parasites, especially the ones capable of manipulating behavior, are likely to completely change the personality of their host, and that people who study animal personality should probably pay more attention to the role of parasites. For example, I suggested that parasites that manipulate behavior might create less consistency in behavioral measurements on the same individuals. They might make these behaviors less repeatable from one day to the next. So in this example here, for instance, if we have two individual animals that are measured, whose behavior is measured repeatedly on different days, for instance, if they are not parasitized, we might expect their behaviors to show quite good consistency. But if they are parasitized by manipulative parasites, I would predict that the behavioral consistency would be lower. And this makes sense because a parasite that alters the behavior of its host, if it's a behavior related to avoiding predators, for instance, it would benefit the parasite to make this behavior more erratic, more random, and less repeatable. It would improve the parasite's own chances of transmission. And the same would apply to behavioral syndromes, or these correlations between different behavioral measurements. I would argue that in some cases, parasites can uncouple, completely break these behavioral correlations. In the example shown here, if we have two behaviors that are measured on 6 different individuals, it's possible that before and after infection, the behavioral correlation will be the same. And perhaps in this case, the value of behavior Z is increased, but it's increased in the same way for all individuals. And it means that the correlation is not really changed. But it's also possible that manipulative parasites could cause a change in one of those two behaviors so that after infection, they are no longer correlated. It could even be the case that the behavioral the manipulative parasites would completely reverse the correlation between two behaviors. Now, why would behavioral manipulative parasites do this? Well, clearly, natural selection could favor this, because in many cases, different behaviors have to be correlated. They have to work together in order to be functional for the host. For instance, if an animal is trying to avoid a predator, there might be a series of different behaviors or phenotypic traits that must work together for the animal to successfully avoid a predator. Perhaps it has to choose a particular background color to hide itself. It has to remain motionless. It has to move a certain distance away from the predator, and so on. If it does not do all of these things in a correlated manner, the anti-predator strategy will not work. Well, for a parasite that is transmitted by predation, it would be a good idea to disconnect these behaviors. So in the past, 7 or 8 years since I've published this paper, it's been cited many times, well over 100 times, but unfortunately most people just cite it as, oh yeah, maybe parasites alter the behavioral syndromes and animal personality. There have been very few studies that have incorporated the potential role of parasites in shaping animal personalities. All these studies suggest that the parasites play a role, but unfortunately there have been very few of those to date. But hopefully people will become more aware of the potential impacts of these parasites. There has been another important study framework to approach investigations into animal behavior, and that's the use of social networks to understand the dynamics within a group of individuals and their social behavior. In the little cartoon shown here, all these green dots represent individual animals, and the lines between them, the arrows between them, represent interactions. The thick arrows represent very frequent interactions. The very thin arrows represent rare interactions. And the direction of the arrow indicates which individuals is initiating a behavior towards which other individual. If we, for instance, think of this as representing the aggressive interactions between animals, this might represent a dominance network, where this individual here appears to be the dominant individual because it directs a lot of aggressions towards others, but no one is aggressive towards this individual. People who study social behavior using the social network approach have to date almost always ignored the way in which parasites might change the position or the role of an individual within these social networks. Imagine, for instance, if a parasite were to infect this individual, and if this parasite is capable of manipulating behavior in a way that lowers the aggressiveness of this individual, well, then maybe the network would look like this instead. This individual, now that it's infected, is no longer the dominant individual. Instead, it is now receiving some aggression from the others, and its position in the dominance hierarchy has dropped down. And this might simply be because it is infected by a parasite that changes its behavior. This is certainly very possible, but always ignored in studies of social interactions. It's not just the position of individuals that might be changed by manipulative parasites, but maybe the structure of the social network overall might also be changed by manipulative parasites. For instance, if we have a social network represented by this little cartoon, where the network represents a single, well-connected bunch of individuals. Imagine that half of these individuals are infected by a parasite that manipulates their behavior in a way that they respond to stimulus differently. For instance, they might show greater attraction to certain environmental stimuli like temperature, humidity, light intensity, for instance. Well, if they do this, perhaps they will move along an environmental gradient so that if they are infected, they would be in one particular portion of the environment, but if they are uninfected, they would remain where they used to be. And there are many examples of parasites that can do this. Many parasites of aquatic crustaceans, for instance, cause their host to be increasingly attracted to light. Therefore, these little crustaceans move to the water surface, but uninfected individuals stay at the bottom of the water. This creates a spatial segregation between two subgroups in the population, and it might lead to the formation of modules, which are subsets of individuals that interact a lot with each other, but only interact a little bit with members of the other module. Once again, the role of manipulative parasites might be very important in shaping the structure of animal social networks, but it has been very little studied to date. So I published a paper a couple of years ago trying to encourage people who study animal behaviors to incorporate the potential impact of manipulative parasites in their studies of social networks. And to date, no one has done it, but I'm pretty convinced that they play a key role in changing the structure of a network. Enough about this broader impacts of parasites. I just want to move on now to the last part of my talk, and it's to focus on the mechanisms by which the parasite is capable of changing the behavior of an animal. Many years ago, the famous scientist Nico Tinbergen, who's one of the founding fathers of the science of animal behavior, insisted that we should study behavior from 4 different angles by asking 4 different types of questions about behavior. We should ask how the behavior develops in an animal from when it is a juvenile all the way to an adult. What are the physiological or genetic mechanism that causes this behavior to be shown in the animal? What is the adaptive function of this behavior? Why has natural selection increased this behavior? In what way does this behavior improve the fitness of the animal? And finally, what is the evolutionary history of the behavior? If we reconstruct a phylogenetic tree of the animals and its relatives, when does this behavior appear in its evolutionary history? And is it shared by other members of its genus or its family? Well, all of these questions have been addressed in the context of host manipulation by parasites. But certainly the aspect that has received the least attention is the study of mechanisms. It's not surprising because it is logistically very difficult to figure out how a parasite inside the host is changing some aspects of the physiology or neurobiology of its host in order to induce a particular behavior. And because it is so difficult, that's probably why few people have studied it to date. The graph here looks at patterns in the study of host manipulation over time. The gray line represents the cumulative number of different combinations of host species and parasite species that have been studied in the context of host manipulation. And you can see that the number of different taxa that have been studied is increasing over time. The red line shows the increase in the number of studies that have looked at the adaptive or the fitness benefits of behavioral manipulation. So in the context of parasites transmitted trophically, that is, transmitted by predation, these involve predation tests, trying to determine whether an infected animal is more susceptible to predation by the definitive host of the parasite. In case you can see this. And you can see that the number of studies that have done these predation tests is also increasing over time, but much more slowly. It's also difficult to do these tests. But the blue line is the one that shows the least increase for many, many years. And this is this looks at the number of studies over time that have looked at the mechanisms by which a parasite can change the behavior of its host. Practically nothing done until the year 2000. And it's only in the last 20 years that there's been an increase in the number of studies trying to understand how parasites can affect their host behavior. Like I've said before, these studies are challenging in many cases, so it's probably why it took quite some time for researchers to focus on that aspect. But now that we've been studying for 20 years, we're starting to understand these mechanisms. So what exactly are these mechanisms? Well, in some cases, it might be that a parasite can alter the behavior of its host in a very simple manner just by being in the right place at the right time. Consider, for instance, this. Why isn't that playing? This should be playing and it's not playing. I apologize for that. Why isn't that playing? Okay, well, the little video should be showing you, this is the eye of a fish, and you can see a parasite in it. And the parasite should be moving because for this parasite to complete its life cycle, it has to be transmitted from this small fish, where it lives in the eye, to a fish-eating bird. And it might be that simply by being in the eye of the fish, the parasite might impair the vision of the fish and make it more susceptible to predation by birds. And in some cases, you can find a lot of parasites inside the eye of a fish. Here you have a fish eye that's been dissected from its fish. I apologize for the shaking picture. It was taken in the field by one of my students who just took the eye out. And you can see that this eye contains a large number of moving parasites. So if this parasite is altering the behavior of its host just by blocking its vision, you can see that it does not have to do anything special. It's in the right place at the right time, it might block vision. And we've actually tried to quantify what the parasite does to vision in these fish. You can see here a cross-section of an eye. So at the dark layer at the back is the retina, where the image is formed. This is the lens. And you can see these parasites caught here in this histological section. And clearly the light that arrives from outside will not reach the retina if the parasites are in front. They will block the light from reaching the retina. So to quantify how often this happens, one of my students used an ophthalmoscope, the same sort of instrument that an optician might use to look at your eyes to see if your eyes are fine. He used that to film the retina from all angles to go around the lens, which is in the way, to get a picture of the retina from different angles and quantify what proportion of the time there is a parasite blocking a particular portion of the retina. So in the composite images that we have here, each pixel represents an area of the retina, and the shade of the pixel, which goes from white to black, indicates the proportion of time that this particular pixel was blocked by a parasite during the video. And he did this on 2 occasions on the same fish, in the morning and the afternoon. So the fish were anesthetized, their eye was filmed for the movement of the parasites. And what he found is that in the morning, each pixel, each part of the retina is obstructed 75% of the time on average, which is quite a bit of time. It's basically most of the time there's a parasite in front of a part of the retina. And the morning is also the time during which the birds feed the most. Actually, it's the only time when the birds feed. So this is the time when the transmission of the parasite is possible. And at that time of the day, the parasites blocks the vision of the fish almost all the time. In the afternoon, however, the parasites move to the sides of the eye and allow the light to reach the retina. They do not obstruct vision so much. And this happens to be a time of the day when the birds are not feeding. So in this particular host parasite system, it seems like natural selection may have favored a very simple response of the parasite. First of all, it goes to the eye and it lives there. That helps. And secondly, it may respond to the changes in the day and night photoperiod and blocks the vision at the time of the day where it is most likely to be transmitted to a bird. So in this case, the behavior of the fish might be changed because it cannot see, but there is no complex mechanism involving biochemical changes in the host. It's simply a physical blocking of vision. In most other host parasite systems, however, it appears like the biochemistry of the host is changed. If you remember earlier, I spoke of these myrmitid nematodes, which grow to a large size inside a terrestrial arthropod, and then they cause it to look for water or water-saturated substrate in order for the parasite to come out where it needs to come out. Well, could it be that somehow the host become more thirsty and they're looking for water? Can the parasite makes the host more thirsty? Well, one of my students quantified the concentration of ions like sodium and potassium in the hemolymph of these crustaceans as a function of the size of the worm they harbor. Obviously, it's only the big worms that cause a change in behavior. And what she found was that in the host with the big worms, there was an increased concentration of these ions and overall an increased osmolality. In most animals, it is known that higher osmolality within body tissues leads to thirst and water seeking. So is this involved in these animals seeking water? Well, we thought that this has to be something that involves a change in the gene expressions of the host. That somehow for these biochemical changes, these physiological changes happening in the host, there must be a change in the gene expressions in the host genomes itself. And that somehow the parasite is capable of reaching into the genomes of the host and turning on or turning off certain genes. Our first attempt to quantify this involved looking inside the brains of these animals, looking for different protein profiles that would reflect the actions of different genes. And what you have here are the results of proteomic studies on the brains of these little amphipods parasitized by the myrmidons. In this study, in red, you have animals that had very big worms. So those are the ones that we expected to show a behavioral change. You have amphipods with small worms. They're not expected to have a different behavior. And then you have uninfected amphipods. Each line represents a different protein, and there are 3 dots for each host because we repeated this in triplicate. And what is shown here on the y-axis is just a measure of, in this case, the down-regulation of certain proteins, just a measure of the relative abundance compared to that of other hosts. You don't need to understand the figure in great detail. What should be very obvious is that for the individuals harboring a large parasite, the protein concentrations in their brains were very, very different from those that were uninfected or those that had a small worm. We did this study also with a different species and a different host. In this case, the host was an earwig. And again, what we found was that for 2 of the earwigs, which had a very big worm inside them, there was a very abnormal profile of proteins in their brains. For the other 2, the proteins look very similar to what we find in the brains of earwigs that have a very small worm. And in these things, these systems, of course, the appearance of the abnormal behavior is something very sudden. It happens over just a few hours. So it's possible that these large worms had not yet initiated the behavioral change, but in this case, it appears that it was initiated. And the role of these proteins is, in most cases, unknown, but because there are databases on the function of proteins determined from other studies, we could identify the functions of some of these proteins. And some of them are involved in neuronal regeneration, that is the growth of neurons in nervous tissues. Others are involved in energy metabolism. So it's possible that somehow towards the end of the infection, when the parasite needs to come out in water, it is inducing its host to become more active through this energy metabolism pathways, because the host needs to move a lot to find water. And it may also be changing the wiring in the brains of these earwigs to make them want to go in water as opposed to avoid the water. Lately, we've been trying to go even one step further in identifying this, and not looking at the proteins themselves, but at gene expression more directly, using a transcriptomic approach by which we measure the number of RNA transcript. RNA is obviously what connects the DNA with protein synthesis. So if we quantify the RNA transcripts in the brains of the infected animals, we can see which genes are particularly strongly expressed or that they are not expressed very strongly. So again, working in this system of the earwigs with the myrmitid nematodes, we expected that there would be different changes in gene expression in the brains of the earwigs over time as their parasite grows from a small worm to a very large worm. Some genes might show a gradual increase or a gradual decrease in their expression over time. Others might show a peak in their expression midway through the infection with no changes after that. Some might peak halfway through the infection. But more interesting for us is this last possibility, finding genes that may show their peak up regulation or down regulation, it doesn't matter, but show a peak change at the very late stages of infection when the worm, as seen here in this CT scan, is very large and ready to come out of its host. If the parasite is going to change gene expression in the host to change its behavior, we expect this to happen only in the latter stages of infection when the worm is large and ready to come out. So doing this study, and this is work in progress, so the results are only preliminary, we found close to 9,000 RNA transcripts that show a change in expression during the course of infection. So each of these little lines here represents a different RNA transcript associated with a different gene. The ones in green indicate that they show an increase in their expression. So in terms of fold change, they are positive. The ones in brown show a decrease in gene expression, so a negative fold change. It doesn't matter whether the change in expression is positive or negative. Here we're only showing the absolute value. What matters is when that change achieves its maximum. And we found that there are close to 7,000 of these RNA transcripts representing 7,000 genes that show their peak change in expression at the very latter stages of infection. And it is really amongst these genes that we might find good candidates to explain the behavioral change in the host when the parasite is big and ready to come out. And indeed, we can, using gene ontology analysis, we can identify the function of these genes using mostly the genomes of other insects that has been fully sequenced. And what we find is that many of these genes whose maximum or minimum expression happens only at the end of the infection, many of them are involved with the locomotion. For instance, they may involve the control of speed of movement, for instance. And again, if the host has to find water, it has to become more active towards the end of the infection process. We also find several genes that appear to have their maximum or minimum expression, genes that are involved in the sensory pathways, whether it's vision or olfaction and so on. And again, this insect has to respond to different stimuli if it is going to search for water, something it doesn't do normally. So we would expect these changes in sensory pathways. But very interesting, we find that several of the RNA transcripts in the brains of the host are actually from parasite genes. These are products of parasite genes that have traveled to the brain of the host, and we're finding them in there. We haven't found very many because at the moment we can only use the genome of the nematode Sinorhabditis elegans as our reference point. But we're in the process of sequencing and annotating the genome of our parasite. So we'll probably find more of these parasite transcripts inside the brains of the host. And again, these are the ones that show a peak change in expression late in the infection process. We don't know the function of all these genes in the nematode parasites that seem to be having an effect on the host brain, but some of them are involved in processes such as messenger RNA splicing or histone transcription. These are all things that control gene expression. So we think that these RNA transcripts, which originate from the genome of the parasite and travel to the brains of the host, are modulating gene expression in the host brain. Now, we have a lot more to figure out as this work is in process to see exactly how this is happening and how particular genes might be targeted. I just want to point out that we're also testing this in a different model system. Remember before I mentioned that there's another phylum of parasites that shows the same convergent life cycle, which also involves growing inside a cricket to a large size and then coming out in water. Well, we're using the same transcriptomic approach with this other parasite system. And you can see here a New Zealand cricket filmed at night, which is infected by one of these large parasites, actually two of these large worms. And you can see one of the worms coming out here, and the other one is about to come out as well. There we go. So the cricket is doing something completely abnormal for a cricket. It goes in the water, it dips its body in the water, and it allows these worms to come out. Why is the cricket doing this? Well, as with the other system, the earwig and the nematode, in this case, we're doing the same sort of thing. We are looking for changes in gene expression in the brain of this cricket to see whether the nematomorph is manipulating gene expression. And since this parasite belongs to a completely different phylum from the nematode, if we find the same genetic pathways involved, it would suggest convergent evolution towards identical mechanisms used by completely different parasites to induce a very similar change in behavior in their host. And just before I finish my last slide, I just want to mention briefly another way we're looking at the mechanisms by which parasites can change animal behavior. Typically, for its entire history, parasitology has often considered the interaction between a parasite and its host as a two-player game. There's a parasite living inside the host. The behavior of the host is changing. It must be because of the parasite. Well, clearly, we have been ignoring the microbiomes of the host and the parasite. Viruses, bacteria, fungi that infect these two individual organisms, and that can also affect the behavioral outcomes for the host. In the last couple of years, we have identified that several parasite species have their own microbiomes, their own communities of bacteria that are not found in the host, not found in the external environment, but unique to the parasites. And these appear to be transmitted vertically. That means that they go from one parasite generation to the next, from the mother parasite to the baby parasite. So that means that whatever is beneficial for the transmission of the parasite is also beneficial for their microbes. So we have now made a funding request. Hopefully we'll get the money. But what we want to do next is to see whether the real manipulative element in this little team of the parasite and its microbes might not be the parasite itself, but maybe the manipulative genes come from the microbes, because these two work as a team. So there is no reason why natural selection would only favor parasite genes that can manipulate host behavior. Maybe the microbes of the parasites also have genes that can help manipulating behavior of the host. So our plan is to characterize the microbes inside parasites that are very good at manipulating and those that are not good at manipulating behavior, and then using antibiotics to change the microbes inside the parasite and then measure if it is still capable of manipulating the host. This is work that is just about to start, so I don't have any results, but I just want to mention that we're also looking at this different approach to how a parasite might be altering the behavior of its host. So I've reached the end. I just want to acknowledge the many people, students, postdocs, and collaborators who have worked with me to study host manipulation over the years. And I want to thank you again for listening to this talk and also for inviting me to present to you today. So thanks again.